How did you begin to look at autism as for research?
Well, as I was saying in the lecture, it was an accident, but I don't believe in accidents. And we were actually doing a different study. We were looking at children with Down syndrome, and we were intervening with some supplements to try to help the children with Down syndrome who also suffer oxidative stress, but a completely different reason. And in order to make a comparison, we have to have control children. And it's very hard to get control children to volunteer. So I asked the moms, the children with Down syndrome, if they could bring in a normal sibling. And they did. And this one mother had twins, and one had autism and one had Down syndrome. So she brought in her autistic child as a control, which was perfectly reasonable. But when we looked at the metabolic profile of these two boys, I couldn't tell the difference. In other words, the autistic child, this was just an accidental finding that the autistic child had a profile that was very similar to the child with Down syndrome, which again shows evidence of oxidative stress. So our first study was simply to find out, was this a fluke? Was this just an accident? Why does this autistic kid look so abnormal? So our first study was simply a follow-up to try to see if in a larger group of kids, did they all have this profile? And so we started out with 10 kids from Buffalo, and they sent, Dr. Paul Cutler sent me their plasma samples, and we analyzed them. And when I saw the results, I frankly did not believe it, because in human studies, you don't see that kind of consistency. You just don't. And so I had to call Dr. Cutler, which was kind of embarrassing, and I said, Paul, I really need you to send me another 10 kids because I'm not sure I believe what we're seeing. Bless his heart, he and Lorette Janik got together another 10 kids, sent me 10 more samples, and it was the same. And so that was the 20 kids that was in the participated in the study that was published in American Journal of Clinical Nutrition. So again, it was kind of a accident that I fell into this work. And as I said, I've been in this business 30 years now, and this is without question the most important thing I've ever done or will do. And I'm very, very hopeful that we'll get NIH funding to follow up on what we've found so far.
Excellent. Now, if money were no object, what kind of research do you think needs to be done?
We need to follow through on our genetic studies. And again, that's going to take at least 1,500 cases and 1,500 controls, because we think that these SNPs, or single nucleotide polymorphisms, that may affect this profile are not going to be the same in every kid. There's going to be at least 3 or 4 of them that interact together that's going to affect the profile. And that is going to take
3 or 4 genes, or what is
3 or 4 different SNPs.
Okay.
One is not going to tell you anything because they're mild, they're small effect changes. So there's never going to be a single SNP to tell you that this is going to cause autism. That's simply not the case. Because they're small effect changes, we think there's going to be 3 or 4 of them that have to interact together to cause a shift in that metabolism that makes them more vulnerable to toxic insult or less able to detoxify. It's not going to be one, that's for sure.
Now, there seems to be a little bit of polarity with genetic versus biomedical research. Could you explain to me, I mean, I personally don't believe that biomedical research insults genetic research at all. Could you explain perhaps how biomedical research helps genetic research?
Yes,
and directs it?
Absolutely. I think we're a step up. We've got a step up in looking at the genetics, because we have what we call a metabolic phenotype. Something shifts those metabolites that we study. Could it be genetics? Because you have this metabolic profile, it gives you big hints which genes might be involved. The other approach, this is called the candidate gene approach. The other approach, which is being by NIH, is a needle in a haystack approach, where you're just looking for what they call linkage disequilibrium. You're looking to see if this gene is transmitted more frequently in autistic families than control families. And actually, it's never a single gene. It's a block of genes, and then you've got to figure out which genes in that block may be important. It's been estimated, and it's agreed, that at least 10 different genes are involved in the genetic component of autism. And that's based on the concordance of identical twins. 80% of the kids, identical twins, have autism. So that means their genes are identical, they both have autism. That is a strong evidence for a genetic component. Siblings are at a higher risk. That's another evidence for a genetic component. But what those genes are, because we think they're small effect genes, it's going to take at least 10 that interact together to create a vulnerability or a genetic susceptibility. Finding those genes with that needle in a haystack approach is going to be very difficult, and it's going to take a long time, especially if there's different genes involved in different kids. And so far, there's been a massive amount of genetic work, and not one gene has been reproducibly identified. So we think that the biomedical approach that we've used by looking at the metabolic profile of these kids, and we see abnormalities, that gives us big hints on where to look, and that's exactly what we're doing. So that's where the biomedical has really led us to the genetic rather than the other way around. And we may be totally wrong, that's what science is, but it certainly is a nice place to stop. It's like a metabolic map to look for the genetics.
Great. Now, there is the issue of whenever these children can't detox, that some people tend to jump on, make conclusions on what caused that. Mm-hmm. The research doesn't point to a cause, does it?
No, in my opinion. Thank you for asking that question. And this is a subtle point, and it's often misinterpreted. And that is the metabolic profile that we've seen that shows that these children, these autistic kids, have low glutathione levels and low cysteine levels that would indicate that they would be less able to handle mercury or a pro-oxidant environmental stress. Doesn't say anything about cause directly, because these children already have autism. So yes, these children who already have autism, they have low glutathione levels. They would be, we could say with some confidence, that they would be less able to detoxify. What we don't know, and this is the important missing part, was that profile there before they were diagnosed with autism? And that we hope to look at if we're funded next year. And that is to look at children who don't have autism, but they are at high risk. And that would be siblings, that would be children who present with speech delays or developmental delays. We can do the profile on those children who are at high risk and then follow them. And if the children that had the low glutathione levels then are the ones that go on to get autism, then we have a much stronger case to say this profile had a lot to do with their getting autism. We can't say that right now because the children that we evaluated already have autism. So all we can say is these kids would be more vulnerable, but we don't know if this profile is a cause or a consequence. It could be simply part of autism that shifted this. We don't know if it was there before. And without that little piece of evidence, we can't strongly say that, that there could have been an environmental trigger to their developing autism. I hope that's clear.